Thank you very much. It's a pleasure to be here today, but I need a little help, so sorry for that. But today I'm gonna talk a little bit about Antarctica as a bioreservoir. It's a research of mine in my PhD. But first of all, why Antarctica? Antarctica can be seen as a kind of archive that will tell us histories from the past, the present, and even the future. Since Antarctica hasn't always been covered by ice, everything hiding beneath they may serve as a record of what the continent was once. Moreover, because remains largely untouched due to limited human presence, the evolutionary process occurs with less interference, and this also provides insight into the present, like how microbial interactions unfold in a relatively controlled sets. In addition, Antarctica is a streaming environment that contains a rich diversity of new enzymes with biotechnological potential, which can be served for the future, for example. Beyond these scientific aspects, Antarctica is governed by the Antarctica Treaty, which promotes collaborative, peaceful research with a strong emphasis in environmental conservation. This is important once Antarctica impact the entire world. So this leads us to the question, what does the continent tell us and how can we preserve this? And quite often it's the microorganisms themselves that reveal the answers. So when we hear about microbial ecology or microbiology in our way, it's very common to us thinking about bacteria. And this is because bacteria which can be studied all the time. But when we think about microbial ecology, this is mean remembering that microorganisms interact with one another and are highly diverse. This way, bacteria can interact with each other, but they also interact with other microorganisms. As example, the bacteriophages, such as viruses that infect bacteria. The bacteriophages are the most abundant biological entities on the planet, and their concentration depends on the ecosystems. So, for example, we have differences between the soil, the mammalian guts, or even the ocean. In this picture, we can see the bacteriophages. And many studies have shown that this virus can modulate bacteria in many ways, like for controlling bacterial by cell lysis or promoting adaptations via horizontal gene transfer through what known as auxiliary metabolic genes. With this field of research growing every day, we have several tools and databases that have emerged to support the advancement of our understanding of bacteriophages. Examples include FASTER, which are used to study phages integrated into bacterial genomes, or even VirSorter, which classify viral contigs from metagenomic data. In addition, there are many databases like Varon, iVirus, and others. In the past 5 years, computational studies of bacterial phages have become even more fascinating because previously it was very common to classify viruses based on their morphology, such as head or tail type. But more recently, the ISTV has updated its classification criteria to allow taxonomic evolution at the genomic level. This change has improved our understanding and allowed the creation of more specific viral groups based on their unique genetic characteristics. So here, for example, we can see in the past classifications, we can call this phase like a Myroviridae. But today, to know this family of his, we need to genetic classification. So today I'm going to share to you some insights from 2 of my studies using sample collected to King George Island in Antarctica, an island that hosts several research stations, including Brazil Station Comandante Ferraz. The first study has the title Humidibacillus stabichis Prophages in Extreme Environments, Comparative Genomics between Antarctica and Spacecraft Strains. So for this, using the Hinterest package, we downloaded 4 genomes of Humidibacillus stabichis from the ECCB database. One of these genomes was from Antarctica, and one other 3 were collected in clean rooms environments used to assemble spacecraft materials. Both environments are considered extreme and have a strong evolutionary pressure on bacteria. Here, we will compare the strains. We observe the Antarctica strain, or lineage 0, is more, have the great number of genes and present a great evolutionary distance from the other ones. So using the FASTER, we can evaluate the prophages in this bacteria, and we observed that the Antarctic strain contains 3 intact phages, 1 degenerated phage, and 1 questionable phage. When I said intact phage, that means that this phage can have integrase, structured proteins, and all with what he needs to be an intact phage. So the generated phage, they have some structured proteins like head or tail, but they don't have the integrase. And questionable is because they have a lot of hypothetical viruses genes, but they don't have these structural genes. So to further investigate the phase of the Antarctic strain, we found that some of them act as recombination hotspots. For example, we can see that phase 3 is very close to phase 2 and phase 4, and this region contains genes that enhance bacterial resilience. Meanwhile, phase 5 was located near other phage genes interspaced with bacterial genes, including antibiotic resistant genes as BCL, which confers resistance to beta-lactamics and wasn't found in the other strains. And finally, in this study, flow cytometry allowed us to identify 2 clusters of activity phage. And in the first, we saw 3 intact phages, but here 2 appears. So we don't know yet if one of the clusters contains 2 phages or only 2 of the total phages are truly isolated. And for the second project, we're going to talk a little about metagenomics analysis of population dynamics between bacterial phages and bacteria in Antarctica hydrocarbon contaminated soils. This work is motivated by the fact that Antarctica has areas contaminated by petroleum due to the expeditions. And one of the most studied approaches to mitigate contamination's effects is bioremediation using bacteria. Whatever, since this is a unique environment with extreme characteristics, sometimes we need to take a step back and understand the microbial ecology of the city to identify the best mechanisms of actions and interactions. Therefore, this study was focused on bacteriophages in these contaminated soils. For this, the study used 5 soil samples along a contamination gradient, from A, the most contaminated, to E, the least one. And from these soils, viral contiguous were extracted by assembling contiguous with mega heat, followed by viral contiguous selection using Reservoir. Taxonomic classification, host prediction, and function annotation that are performed too. So about 260 contiguous were identified across all soil samples. Most contiguous had no predicted hosts, and most were classified only at the class level. Like in this case, Caldoviricites were very common to bacteriophages. But for the contiguous with predictive hosts, we created this image to show the virus taxonomy alongside their respective hosts. So there is the virus, and here the hosts of this virus. We can see, for example, that the bacteria Gordonia had the highest number of associated contigues. And interestingly, some of these contigues were classified into different genes, such as stomatovirus, gallonovirus, and some caldoviruses without genes. Additionally, this image allows us to observe the potential hosts of viral contigues grouped by each soil sample, and soil B had the most viral contigues and the highest number of contigues with predictive hosts. Another interesting fact is the fact of these predictive hosts are bacteria classified as potential petroleum degraders, such as Gordonia, Pseudomonas, Haustomonias, and Rhodobacter, among others. This is still a characterization study, but it begins to provide curious insight into how viruses and bacteria interact in extreme soils like those in Antarctica, which are even more complex to the petroleum contamination. So I would like to say a special thanks with all people including this project, especially to my advisor, Diogo Giralevicius. And that's it. Thanks a lot. No, we have the 4 samples, all downloaded by the NCBI. So 1 sample is from us, the lab, but 3 is from the NASA, who put the samples in the NCBI and we downloaded to compare with our sample too. The spacecraft from NASA, yes. Spacecrafts.
And where did the spacecraft go?
I have no idea. This I don't know.
So, do you think there's a similarity in the ecology with the spacecraft and Antarctica?
Actually, we found about the phage, just about the phage, our gene-generated phage has some similarity to the generated phage found on the spacecraft samples. So, for example, our samples just have the intact phages and 1 degenerated phage. The other samples has 1 degenerated phage, which with similarities with each other, themselves, and with the Antarctic strain. I don't know if I can explain more.
So, there's some similarity in the phage
Yes,
population.
yes, there is.
And the degenerate ones, is there gene sharing?
I'm sorry, I can't hear you. What?
So for the degenerate phage.
Okay,
They don't have the integrase,
they don't have any.
but something else has an integrase in
they,
the cell.
they have just the head protein.
Hmm.
They generate. And somehow, someday they had the integrase, but
Mhm.
today in this sample they don't have anymore.
Okay, but was there a different phage in the same cell that encoded the integrase?
Between the generated phase, seems like the same,
Hmm.
but we don't know for sure.
Thank you.
Thank you.
So I'm going to actually follow up on Danny's question a little bit.
Of course.
I'm curious. The thing that went through my head with the spacecraft thing is, I wonder if it's just radiation exposure.
Oh,
And
no.
did you look at other parts of the bacteria to see if they had a lot of changes relative to the
Yes.
other strains?
Actually, the principal mean here is that the spacecraft material is assembled in the clean rooms that is very, very extreme pressure in bacteria. So it's not about the radiation, but the cleaning,
Oh,
okay, the rigorous cleaning. But the both Antarctic strains and spacecraft strains have genes to this kind of stress. So in Antarctica, some phase have the genes for cold stress, for example, but all of them, independent of the phase, have radiation protection, for example, in Antarctica too.
Can I ask?
Go ahead, please.
Hi. So you inferred from the number of phage contacts, you inferred the bacteria that you found and also quantified that. Did I get that correctly?
In this, can you talk a little?
So you looked at the phage contacts that you collected, and you then inferred from that
From
which
the
bacteria
contacts.
you thought were out there, and also in the frequency of those bacteria, right? There was a quantification step. Is that did I get that right? There was a slide. Do you want to show that?
Actually, here in this and
Yeah,
this
that's right.
slide.
Yes, yeah.
Yes, here actually is about the contiguous we assemble. So it's about the contiguous that we classified as virals.
Yeah.
And from this, we can presume what this host is.
Yeah, and so my question is, doesn't that don't you need a sort of viral load number in order to draw the fatness of your edges in this graph?
So,
Well, like for example, if the viral load in Gordonia is very different from,
yeah,
then somehow the fatness of your edges doesn't seem to, or
Because-
yeah, it needs to be normalized, right?
because it's different virus in this case. We had about 200 contigs in the genome. And when we did the host prediction, we had the Gordonia appears most of the times, but different virus. So we had, for example, Caldovirus without genes, but gallonovirus, which is a virus that infects Gordonia. That's okay.
Okay. Next, thanks, speaker again.